So welcome to Respiratory. We only have one video this time, but you do have a series of handouts on different respiratory disorders to go through. They're fairly simplistic. There are also associated quizzes, so please have a go at those. And then in this video, what we'll look at primarily is about infection. So we'll look in terms of acute infections, but also one chronic bacterial infection. And then, as usual, we'll get on to the applied pathophysiology, where we look at the signs and symptoms that are common to many respiratory disorders. So let's start with acute infections then. Hopefully you've read the pathogens handout before we go through this. If not, please stop now and go and read it. So we'll start by talking about viruses. As I say, hopefully you have looked at the handout beforehand and you're aware that viruses do come in different shapes and different forms. So viruses can contain DNA or RNA as their genome. That DNA or RNA can be a single strand or a double strand, and that single strand can be a sense strand or an antisense strand. And this all determines exactly how they replicate, what they need to do to replicate. They also contain a few basic proteins such as polymerases. We'll talk about what the polymerases do in a minute. But they don't have their own organelles. So they don't have ribosomes, they don't have a nucleus, they don't have anything that can enable them to replicate independently. So therefore, they have to get into host cells in order to replicate, to use the proteins that are naturally within your cells. They're surrounded by a tough protein coat that's called a capsid. Here you can see the hexagonal structure is the capsid. Many of them then have an additional envelope around the outside that's actually a phospholipid bilayer. And generally this is formed from your plasma membrane when the baby viruses, if you like, bud out from cells after replication. They take a little bit of your plasma membrane with them to wrap around their capsid. Some of the proteins on that envelope are receptors because in order to actually get into host cells, these viruses then have to bind to those host cells to enable them to get in, to be endocytosed. A complete virus outside of a host cell is called a virion. So viral replication then. Viruses will bind to host cells. So they have various receptors, they have proteins on their plasma membrane, you have various receptors, and their proteins on their envelopes may fit those receptors. Different viruses bind to different receptors. So they bind to lipoprotein or glycoprotein receptors on the host cell. They then are fused with the host plasma membrane, or they're endocytosed via receptor-mediated endocytosis. If you remember, this is the formation of the clathrin pits and all the rest of it, quite an involved process. Or they can just fuse and enter. Once they're in the cell, they lose the capsid, so they uncoat. Because they're removing their coat, this capsid, they're exposing their genome, their RNA or DNA, so that it can then be used for replication. As we said, the mechanism of replication depends on whether that viral genome is RNA or DNA, whether it's single or double strand, or whether the single strands are sense or antisense. So, as a nice simple example, let's look at the replication of a sense single-stranded RNA virus. So this is often referred to as a positive strand because it's the sense strand, the antisense strand being the negative strand. So this is a positive or sense single-stranded RNA virus. And sometimes you may see it written with a little plus, a couple of small s's, and then RNA. So RNA viruses with a single sense strand, this is really straightforward because they're translated via the ribosomes within the host cell, just like host messenger RNA. That's basically what they are. If you remember the sense strand that goes through the ribosomes in your own transcription, translation, is a sense strand. So the sense strand of the virus can be put through the ribosomes in exactly the same way as your own messenger RNA is. This enables it, because of the codons on that single sense strand, it enables it to synthesize the proteins that it needs for all the little babies it's going to make. So all the polymerases, the capsid proteins, that sort of thing. So the single or positive sense strand comes in from the virus. That's then transcribed to form a polypeptide chain, again, using your own amino acids, but to form a viral protein. So the viral proteins are then needed for the capsid and for the enzymes of the new virions. In addition to this, of course, all the new virions are going to need their own positive or sense strand so that they can go off and they have their own genome. So what our virus that is replicating has to do, it has to make a template for that. So as we said, viruses have other proteins, other simple proteins. One of those is an RNA-dependent RNA polymerase. Now, this RNA-dependent RNA polymerase copies the original viral sense strand to give a complementary, a paired, antisense strand. This is the viral template, if you like, to make more sense strands. So viral RNA-dependent RNA polymerase works its way through the single positive sense viral strand, and it makes this template that is antisense by pairing ribonucleotides. This antisense or negative strand can then be used to make lots and lots of new sense strands. Those new sense strands are going to be the genome of all the new virions. Because of all of this, this is where we get this problem with antigenic shift and drift. And it's particularly common, for example, in flu viruses. Because there's a high error rate in doing this RNA replication using this template, then we get differences. Subtle differences can arise quite easily. So the viral RNA is likely to mutate And this gives us all those virus variants, the antigenic shift and drift. It might be a difference in a binding protein or anything else in terms of that virus that will then be replicated again. So in other words, we've got the same virus, but now we've got a variant. So slightly more complicated is the replication of a negative or antisense single-strand RNA virus. And again, this is denoted as a minus 2 small s's RNA. In this case, before it does anything, the virus has to use RNA-dependent RNA polymerase to create a positive or sense strand. Once it's done that, of course, it's at the same point that we had with our positive, our sense strand RNA virus. It has something that can be used in the host ribosomes as messenger RNA to make those first proteins. DNA virus replicates generally inside the nucleus of the host cell, and it uses the host cell proteins. It's quite a complex process, but it also uses your RNA polymerase. And what happens is you get copies of the viral DNA made, and you then from that can make the viral version of messenger RNA, which can then be transcribed, sent out, off to the ribosomes, translated, and into viral proteins. So a slightly lengthier process. Once all that's gone through, depending on whether it's an antisense, sense, single strand, double strand, RNA, DNA, once that genome is replicated and the host cell, the virus has caused the host cell to synthesize some capsid proteins, some polymerases, all the things that new viruses need. That's all put together, and the new virions are formed within the host cell. So the host cell had one virus in, now it's got mummy virus and lots of babies. Those virions, when they're ready, they're formed, they leave the host cell, maybe by causing lysis or by budding from the plasma membrane. And as we said, that envelope comes from your own host cell plasma membrane. So they're taking a little bit of membrane with them. And you can see here, they're doing it in such large quantities that yes, they are likely to cause lysis. This budding, there's lots of virions coming out from one of your host cells, taking lots of plasma membrane. They're going to leave holes. And that cell will generally die. Reverse transcription is worth talking about because it's how HIV replicates. And it's a complex, but it's a very sneaky process. In reverse transcription, so these are called retroviruses, such as HIV. There are others are available. They contain two identical single-sense RNA strands linked together, sort of end-to-end, if you like. The viral RNA-dependent DNA polymerase in this case, also known as reverse transcriptase, transcribes the viral RNA into DNA. That viral DNA, so this is a whole new sequence of codons, unfamiliar to you, but absolutely wonderful for the virus. This viral DNA is then bonded into the host DNA using an enzyme called integrase. So it's actually inserted into your DNA. And this modified DNA is then used to produce viral proteins in the same way as your DNA would produce your proteins. So we can see through all of that these viruses are going to have a very damaging effect on host cells. So they cause death of the host cells through lysis, when large numbers of these new virions bud out and are released. Also by immune-mediated lysis or apoptosis, if you remember back to immunology. If you have an immune response, you have things like membrane attack complex. If you have an adaptive response, you have cytotoxic T cells. You have lots of things that do in their own right as inflammatory mediators or inflammatory processes mediate lysis or apoptosis. The other thing is having a potential intrinsic death pathway. So if there's a cessation of the normal intracellular activities because it's full of virions, there are lots of transcription, translation going on, they're using all the host proteins, then that could potentially stimulate mitochondrial stress and activate the intrinsic death pathway, leading to apoptosis. Of course, lysis and apoptosis are further going to stimulate both the innate and then eventually the adaptive immune response due to the secretion of colony stimulating factors, all the cytokines, TNF-alpha, the interleukins, the prostaglandins, degranulation of mast cells. So we're going to have a full-on inflammatory upgrade going on in response to the invasion of these host cells, in response to lysis and apoptosis. So, okay, some specific mechanisms in terms of the immune system that are particularly useful in a viral infection involves both the innate and then eventually the adaptive responses. In terms of the adaptive response, of course, if we have cytotoxic T cells, these cytotoxic T cells have maybe been activated against viral proteins, so they are antigen specific for some of the viral proteins. If you remember, all cells have MHC1. When those cells are healthy, they're not infected, they're displaying normal proteins. When they become infected, they will switch those, bring those down, and put up some new ones. And these ones might be displaying viral proteins. Our little virus is busy replicating. Oh, let's steal some proteins, stick them up on the membrane, and an MHC1. If those viral proteins have already activated cytotoxic T cells, then they will come and bind to that via the T cell receptor. Also, T helper cells, of course, go around secreting lots of cytokines, TNF-alpha, interleukins, other chemokines. Chemokines, of course, promote chemotaxis. And ultimately from chemotaxis, you have phagocytosis of potentially free virus or the killing of infected cells. They're then going to, of course, activate more adaptive responses. If we activate our adaptive responses, ultimately those T helper cells will help to activate B cells, B lymphocytes, B cells. They, of course, become plasma cells and secrete antigen-specific antibodies, immunoglobulins. These antibodies will bind to free virus. If you remember, this is neutralization. So they will bind to free virus and they will block it either from binding to a host cell, so it can't actually get into a host cell to replicate, or they could potentially stop it uncoating once it gets into the host cell. Activation of complement, of course, is another role of antibodies. So these could bind to free virus and then to the Fc end, that's via the Fv end. The Fc end binds to C1, complement 1 from the liver, which then, of course, goes through those whole pathways and we get things like membrane attack complex that can result in the lysis of infective cells. Of course, if it does result in the lysis of infected cells, then we end up releasing the virus, in which case we then have to deal with it. Also leads to opsonization, of course. And then there's natural killer cells. Natural killer cells, if you remember, antibodies in antibody-dependent cellular cytotoxicity can bind to a natural killer cell via the Fc, which leaves the Fv to bind to the antigen. When that Fv binds to antigen, the natural killer cell then releases its perforins and granzymes. It becomes a bit like an antigen-specific cytotoxic T cell due to an antibody binding to it. And as we said, there's also, if you're activating complement, we have the potential for opsonization, not only from C3b though, but also from our antibodies. They can opsonize virally infected cells or free virions, leading to their phagocytosis. Interferons, of course, are really quite useful in terms of intracellular infection, in terms of viruses entering host cells. So we're talking about interferon alpha and interferon beta. These are type 1 interferons. There are different classifications. They're secreted from cells when those cells are infected by a virus. And this usually happens fairly quickly. We're talking about sort of, 12 hours up to 2 days, this happening. So our little virus infects the cell, and of course our little virus then replicates, and it releases its free virions. So far so good for the virus, not so good for us. But when that happens, interferon alpha or interferon beta, these type 1 interferons, are synthesized by the infected cell, released from the infected cell, and then bind to an interferon receptor on a neighboring or an adjacent cell. So happily, there's virus replicating away. The cell, meanwhile, is doing a sneaky backdoor job by releasing interferons. These interferons, when they bind on the neighboring cell, stimulates kinases. So we're talking a second messenger system involving phosphorylation. Remember, kinases always phosphorylate. This triggers the synthesis of antiviral enzymes by that cell, the neighboring cell. The enzymes go off and they block double-strand RNA interactions. In other words, our RNA genome from the virus can't make a template, so it can't replicate its own viral RNA. If you can't do a double-strand interaction, you can't replicate that RNA genome. So they block that happening. So even though the virus has entered the neighboring cell, that virus cannot replicate. So it's not going to be released, it's not going to infect lots of other cells. Unfortunately, in some cases, this may also impact on that host cell's ability to control its own protein synthesis. There's usually no effect in non-infected cells for various reasons. But infected cells, it may actually cause apoptosis of that cell. But, okay, we've now lost another cell, we've now lost two cells, these two neighboring cells. But at least we're not releasing virions to infect lots of other cells, and they all die too. So it's a bit like the needs of the many outweigh the needs of the few. The other thing that interferons do, they increase the expression of MHC1 within the host cell. So that's great because what happens is if you have more MHC1 sticking out holding that viral antigen, there's a greater chance that that cell will be sent into apoptosis by an activated and antigen-specific cytotoxic T cell. And talking of cytotoxic cells, natural killer cells are also activated when interferons are released into the extracellular environment. So we've got lots of killing mechanisms going on. Those killing mechanisms are going to reduce the opportunity of the virus in this immediate area to find a host cell in order to replicate. So that was viral infection, a general overview. Different viruses have different receptors, have other different mechanisms, but that's a general overview of how viruses replicate and how the immune response can defend against them. Bacterial infection, obviously, apart from talking about a totally different pathogen, we might need different mechanisms to deal with. In the case of bacteria, we've not only got to deal with bacteria, and actually there are a couple of viruses that also have toxins, but bacteria secrete toxins, and those toxins can have a destructive effect in their own right. So we've got the presence of bacteria, we've got the presence of toxins, we've got tissue damage because of that. We are going to get an innate immune response. Of course, we have tissue macrophages, and here we're talking about respiratory. So we're talking about alveolar macrophages. So our little bacteria gets past all the other defense, the barrier mechanisms, mucus, mucociliary clearance, all of that gets down into the alveoli. Hello, I'm a tissue macrophage. I'm going to phagocytose you and get rid of you. So our tissue macrophages, of course, are already there. They're sitting there waiting. They phagocytose free bacteria or any cell debris from damage that the bacteria have caused. When they do that, of course, they always release lots of chemical mediators like colony stimulating factors, cytokines, prostaglandins, chemokines. These are all umbrella terms. So we're talking about a lot of chemicals being released here, when a macrophage, or a dendritic cell for that matter, phagocytoses. In addition to this, any infected or damaged cells will also secrete colony-stimulating factors, cytokines, prostaglandins, and chemokines. So we're building up a bit of a soup of inflammatory mediators. And just to top it all off, mast cells can be activated, and mast cells can actually secrete inflammatory mediators, and they can degranulate, releasing histamine. So we have sort of a bit of a bog-off deal. We have two for the price of one with mast cells. And they can be activated in a variety of different ways. When they do, they secrete and they degranulate. Degranulation releases histamine, which of course is one of our main vasoactive amines. The colony-stimulating factors, meanwhile, remember this is a bit of an umbrella term, we have quite a few of them. More commonly, we have granulocyte colony-stimulating factor, or macrophage colony-stimulating factor, or granulocyte macrophage colony-stimulating factor. There are quite a few of them. And of course, the cells that they stimulate, the clue is in the name. So these colony-stimulating factors go off to the bone marrow and they stimulate leukocytosis. So if you're doing a blood test, the white cell count at this point is going to go up. In terms of what's happening in the body, lots of these leukocytes, and maybe neutrophils first, then macrophages, are all, or monocytes in circulation, are all being released from the bone marrow. So our little tissue macrophage down in the alveoli is calling for some friends to come and help him. So the circulating neutrophils and eventually monocytes are able to emigrate into the tissues. You remember this is all about roly-roly, put a foot through, diapedesis, stick to the cell adhesion molecules first so that you can get your foot through, and eventually emigrate through those gaps into the tissues. And they're able to do that because of vasoactive changes. So we've got vasodilation, remember, which slows the blood. We've got increased vascular permeability because our endothelial cells all shrink, giving us bigger gaps between them. And this is all stimulated. We always talk about histamine as being one of our main vasoactive amines, but also by prostaglandins, by the cytokines, and by a few other mediators. These all cause vasoactive changes. Those vasoactive changes will facilitate emigration of the cells that we have just released into the circulation due to the colony stimulating factors. Their entry, their emigration is also facilitated by the cell adhesion molecules. Cell adhesion molecules, amongst other things, are stimulated by TNF-alpha and interleukin-1. So if you remember, their expression is stimulated on our endothelial or epithelial cells, but also on the white cells themselves. We have, like our little sticky Velcro hooks and eyes, our little loops that they can stick to. So, our emigrated cells are guided to the target by this chemotactic gradient of chemokines secreted by damaged cells, by phagocytes already in the area. Once neutrophils get in, their role is partly to degranulate. When they degranulate, they release proteases and lysozymes, so toxic enzymes that will break things down, including the target, but also, unless closely controlled, our own host cells. They can phagocytose, but they tend to die very quickly when they do. And they have these funky neutrophil extracellular traps, nets. So they chuck out chromatin from their nuclei. The chromatin has PRRs in it, it's very sticky, it grabs hold of pathogens, debris, and then those proteases and lysozymes within the net then break those targets down. Monocytes, as we said, can become macrophages. They can also become dendritic cells. Once they do, they phagocytose bacteria by binding their pattern recognition receptors to PAMPs or to cell debris via DAMPs. All of these recruited immune cells then release more colony-stimulating factors, more cytokines, more prostaglandins, more chemokines, upregulating the whole response again. So we get an upregulation of the innate response and inflammatory processes. The innate response, then, if you remember the link, is an antigen-presenting cell. So phagocytosis results in an antigen-presenting cell that's presenting specific antigen to lymphocytes. That then activates our adaptive response. So our lymphocytes, when they're activated, become antigen specific. So T helper cells secrete interleukins, other chemical mediators that help to upregulate the innate response. We also get cytotoxic T lymphocytes, TC cells, and we get the activation of B cells, B lymphocytes that then become plasma cells and secrete antibodies. Our cytotoxic T lymphocytes destroy the infected and damaged cells or any extracellular pathogens, whatever they have been made specific to, whatever antigen they are now specific to. They will bind and destroy. B lymphocytes, as we said, become plasma cells and they secrete antibodies. But all of them, if you remember, the key thing, other than being antigen specific about the adaptive response, is that we make these memory cells. So some of the cells that proliferate, the T and B cells, will go out and deal with the problem at the moment, but some of them will get stored away in lymphoid tissue, ready for subsequent infections with that particular antigen. So that was a sort of general view, really, and quite a useful general view in terms of any inflammatory response. It was a quick revision of everything that we did in immunology. In terms of bacterial infection, we also have some specific defenses. And what do you think would determine this? We've already talked about tissue macrophages. Now, tissue macrophages vary depending on what tissue we're in. In other words, the localization of the infection. So, the specific bacterial mechanism of action also determines the amount of host cell damage. So that depends whether it has toxins, its defense mechanisms, and also whether it remains outside the cells extracellular, or some bacteria can actually enter cells. And as we said, the location as well is going to determine the specific responses. So we're talking here about which tissue macrophage is involved, that any localization, whether it can be walled in, or specific defenses such as mucus, pH, things like that, things that are specific to the area. Flushing out, for example. In the lungs, of course, our local macrophage, our tissue macrophage, are the alveolar macrophages. Clearance, we also have, of course, mucociliary clearance. So in addition to general immune responses, neutrophils, natural killer cells, and endothelial cells also secrete something called defensins. Defensins are a bit like membrane attack complex. Not the same structure and not part of complement, but they do create pores in targets. So defensins create pores in bacterial cell walls or in viral envelopes. And exactly the same thing will happen there. We'll end up with lysis. We also can activate complement. So we have, if you remember from your handout, you have CRP can activate complement. We know antibodies can activate complement. And the various complement proteins then degranulate mast cells, they opsonize, or they form membrane attack complex. So let's run through an example. So if you're asked for an example in a question, you have to be able to name it in full. And technically, any name should be given in italics, but we're not that pedantic, but it should be given in full. So we're going to talk about Streptococcus pneumoniae, otherwise known as pneumococci. Now, they have their own little defense mechanisms, and they are quite specialist, really. This is possibly why we've chosen this one as an example, to show that we need to have different defenses for different mechanisms of action. So streptococci, or Streptococcus pneumoniae, or pneumococci, release hydrogen peroxide, and they do this during this autolysis. This autolysis, if you remember, is this central clearing where the young, they die quite quickly as they spread out. When they die, they release hydrogen peroxide. Of course, hydrogen peroxide, not a nice chemical to have around. It's going to cause endothelial damage. That's then going to stimulate cytokine and chemokine secretion and potentially the production of nitric oxide. So what's going to be the result in terms of our body to that? They also secrete pneumolysin during autolysis. Pneumolysin is another one that creates pores in host cells, but this one's being produced by the bacterium itself. Creates pores in host cells, also recruits large numbers of neutrophils. Now, anything that secretes large numbers of neutrophils is known as pyogenic, not pyro, pyo, P-Y-O, genic, because they create pus. If you recruit large numbers of neutrophils, neutrophils die very quickly, therefore you end up with a lot of dead neutrophil, a lot of debris, a lot of necrotic cell, and this gives pus. Pneumococci also have a capsule that resists opsonization by both C3b and antibodies, immunoglobulins. This is going to reduce its chance of phagocytosis. In terms of significance, just think about that. This is going to be a lot harder to clear up through phagocytosis, one of our key defense mechanisms. If you can't phagocytose it, it's also going to be very difficult to produce antigen-presenting cells. It can also go intracellular. So it can use receptor-mediated endocytosis. It will bind to a pre-existing receptor, become endocytosed, and enter endothelial cells and exocytose at the basal surface. Remember, this is our movement through the cell, transcytosis. And the significance of this, it's going to get away, isn't it? If it can get through endothelial cells, it can get out of the circulation, it can get into the tissues, and it can start to move through the tissues out of the reach, potentially, of any defense mechanisms. Okay, so that was just an example really of a particular pathogen and how the mechanisms that they defend themselves with or that they damage our cells with can make a difference in terms of our ability to defend against it. Talking about defending against things, of course, we've always thought, well, we've got antibiotics, but we know now that that's not quite so reliable. We've got resistance is growing. We've got increasing resistance to more and more antibiotics. And the problem with this is that we went through a phase, we discovered antibiotics, we discovered penicillin, great, lovely. We put lots of research in and we made lots of new antibiotics. Well, that will keep us going forever. The problem is that we're not finding new antibiotics, but meanwhile, the pathogens are increasing their resistance by mutating, by various other mechanisms to those antibiotics. And we're not talking just about bacteria. This is also becoming true of antivirals as well. We are developing resistance to antivirals. There are three possible mechanisms really. In some cases, the pathogen is able to degrade the antibiotic by releasing its own enzymes that degrade it. Another possibility is alterations to the binding sites on the bacteria that the antibiotic will bind to. If we have a slight difference in a particular receptor that this antibiotic uses, that antibiotic may no longer bind and therefore it can't be effective. And possibly changes in the membrane permeability to antibiotics, depending how that antibiotic works. And of course, all these mechanisms, all these defense mechanisms against the antibiotics, those bacteria are going to survive, or viruses are going to survive. If they survive, they can go on and replicate. So we've got a whole new massive generation that is resistant to our drugs. And the World Health Organization have said that antimicrobial resistance is one of the top 10 global public health threats. And they said this fairly recently, so they're including COVID in there somewhere as a possible threat, but still this is within the top 10 of public health threats. And I've given you a link on Moodle to the World Health Organization's antimicrobial resistance page if you want to read more, or if you want to use it to demonstrate to a colleague why they should not be prescribing in a particular situation where it's not especially indicated or necessary. Which brings us on to chronic infection. And I've intentionally missed the S off there because we're only going to talk about one. We're going to talk about tuberculosis. Before we do that, what we need to talk about is the word chronic, really, in terms of inflammation. Chronic inflammation varies from acute, not particularly in time frame, more about the mechanisms that happen as a result of it. So chronic inflammation is characterized by this concurrent tissue damage and repair. So you get a mixed morphology. So you might have tissue repair going on alongside ongoing damage. That damage then goes into repair and you've got another area of damage. So you've got all these things happening all at the same time. The damage itself is facilitated by things like proteases, by our free radicals, reactive oxygen species, lysosomal enzymes, anything that is going to damage the tissue because we've got an inflammatory process going on. The repair part, of course, a part of inflammation is that we then have to, once we've cleared everything up, we have to repair the tissues. So repair is facilitated by various growth factors. And our sort of classic ones are fibroblast growth factor, secreted by, yes, you guessed it, fibroblasts, and also vascular endothelial growth factor, yes, secreted by vascular endothelium. Both of these will stimulate cell proliferation and angiogenesis, particularly in the case of vascular endothelial growth factor. If we're going to repair tissue, we need a new blood supply. So we need that blood supply to bud off and grow into the new cells that we're laying down. If, however, the tissue loss is too great or too sustained, and this is the thing with chronic inflammation occurs because we keep stimulating the inflammation. Either we have an obstructed duct, we keep doing the same thing over and over that causes inflammation, and it never manages to resolve because we keep stimulating it. If tissue loss is too great or too sustained over a period of time, or the cells that we're damaging have a low ability to proliferate, say for example neurons, repair may result in an area of fibrosis. So we might end up replacing those active cells with collagen, disorganized collagen fibers. Normally, we would lay down collagen fibers for the cells to grow into it. But if those cells don't grow into it, that collagen never gets broken down, and we end up with an area of scarring or fibrosis. In chronic inflammation, as you might expect, there's also a prevalence of macrophages because, of course, neutrophils come out in the first response. So later on, we tend to get monocytes, so we're going to have more macrophages. And fibroblasts, because, of course, we're attempting to repair the bit that's just been damaged. The chemicals involved, or the whole range is involved really. But the really sort of three big bad boys, as I call them, are TNF alpha, interleukin 1 and interleukin 6. And these cause a lot of the systemic problems as a result of chronic inflammation. These cytokines are our main cytokines causing all the problems in a chronic inflammatory state. Of course, we have an extended period of inflammation. No one's going to say you've got chronic inflammation after 3 days, but there is no specific duration. It is really more or less judged on this mixed morphology of damage and repair rather than on time, even though chronic comes from the Greek chronos for time. So tuberculosis, to bring it back to respiratory, TB is a specific type of chronic inflammation. So we can have a more diffuse chronic inflammation, as we know. We can have joints that are inflamed for many years. We can have a low-grade chronic inflammation if we overeat or we're overweight, and that has adverse effects systemically. But TB is this granulomatous inflammation. It has this key characteristic of forming granulomas. Other conditions do cause this, so they can be infective or non-infective. So of an infective variety is something called cat scratch disease, Bartonella henselae, where you form granulomas. Another key one, leprosy. And if you've seen in films, you get pictures of people with tubercles hanging from their axilla or wherever, characterized as leprosy. And these are the granulomas. Syphilis is granulomatous, sarcoidosis, Crohn's disease, and even retained foreign body. So, for example, someone has a piece of retained suture or any foreign body, it will form a granuloma around it. Another example is food aspiration. These all tend to develop this sort of granulomatous morphology, if you like, where we have these granulomae. So in tuberculosis, because we're looking specifically at TB, again, as I said, you need to name your pathogen in full. Mycobacterium tuberculosis. This one is aerobic. You might expect it to be. It enters via the lungs. But it's also very slow growing in culture, and it's very resistant to drying. So the problem is, if someone exhales it, spits it out, whatever, it gets into some dust, then it can sit there and survive for years. Then all of a sudden you get a bit of a breeze, blows the dust up, you inhale the dust, and there you go. It can then start to become more active. So when we do inhale it, and we are only talking about inhalation here, we'll talk about how it reaches other parts of the body a bit later. Always inhaled. On inhalation, mycobacteria get down into the alveoli. There's our little alveolar macrophages. Oh, hello, you're right, I'm having you, mate. I'm going to phagocytose you and get rid of you. And there you've got a lovely picture of a macrophage phagocytosing mycobacteria. That was actually given to me by a student several years ago. But the problem is mycobacteria can be phagocytosed. Normally that would be the end of them, wouldn't it? Because along comes our lysosome, fuses with our phagosome, we get a phagolysosome, and the contents of the phagosome get broken down. Mycobacteria have this coating, so they actually stop the lysosome from fusing to the phagosome. Therefore, of course, mycobacteria can happily sit in the phagosome without any nasty toxic chemicals coming to kill it. So it's not destroyed by lysosomal enzymes, and it can end up surviving in our phagosome, in our macrophage, for ages. So, of course, our macrophage did manage to phagocytose it. As soon as it phagocytoses, remember, the macrophage then secretes its cytokines, prostaglandins, macrophage colony stimulating factor, and various chemokines. So all our normal inflammatory mediators that are going to give us an inflammatory response. That inflammatory response involves leukocytosis, emigration, chemotaxis, and all of this means that we end up with a lot more macrophages recruited to the area. They see some more mycobacteria and go, oh, right, mate, I'm going to have you. And they phagocytose the mycobacteria. Of course, they then secrete more prostaglandins, cytokines, colony stimulating factors, etc., calling more macrophages to the area. Those macrophages come in, right, mate, I can see you mycobacteria, I'm going to have you, I'm going to phagocytose you. What we end up with is a big ball of infected macrophages, primarily infected macrophages. This dense ball of infected macrophages that can't get rid of the mycobacteria within them then forms the granuloma. Along come our fibroblasts. After all, this is chronic inflammation, so we're going to have some fibroblasts. Our fibroblasts are recruited. They're activated by various cytokines in the area and growth factors. They secrete their collagen and they cause fibrosis. In this case, that fibrosis walls off the granuloma. So it starts at the periphery because that's where our fibroblasts first land. Oh, yeah, let's secrete loads of collagen around this. And they end up walling it off. Well, that's all very good because we've now contained our infected macrophages. So they wall it off. And what we've actually got is a non-progressive or latent tuberculosis. It's just sitting there, walled off quite happily, can't spread anywhere. We've walled it off with our fibrosis, our collagen. In some cases, the fibrosis can continue and it can virtually fill the granuloma with this scar tissue, with this collagen. And what we end up with is something called involution or resolution, which means that we might have, yes, a small scar that might or might not be seen on X-ray, but we have this little patch of fibrosis where we had TB. However, within our granuloma, so we might have walled it off, but within the granuloma, those macrophages, they don't just sit there forevermore with mycobacteria in them living synergistically or in a nice neighbourly way. Unfortunately, the mycobacteria cause various changes to the macrophages. One of the changes that can happen, and this isn't really terribly significant in terms of the course of the infection, but it is quite interesting in terms of the histology of the granuloma. These macrophages can form multinucleate giant cells. So if you remember, our only multinucleate cell normally are skeletal myocytes. In this case, we have a pathological multinucleate cell. And they do come with various names, giant cells, Langerhans cells, various things, but they're all a variation on the same thing. And it's suspected that you get these multinucleate giant cells because multiple macrophages all cluster together, tight up against each other, try and phagocytose the same mycobacterium. And when they do that, they end up fusing. That's the theory anyway. And the role of them isn't really known. As I said, that in terms of the progression, they're not really that significant. But they are in terms of seeing the histology. They do also tend to die quicker than unfused macrophages. And this is really where the significant thing comes in, is that these giant cells are not much good in the usual role of the macrophage in phagocytosing. Another thing that can happen to the macrophages is they turn from being phagocytes to being primarily secretory. And this happens in a lot of macrophages really after a period of time. They tend to switch to become more secretory. There's a genetic signaling pathway, etc., that changes the genetic expression in that macrophage. When they do this, they become these elongated, sort of slipper-shaped macrophages that are called epithelioid cells. And these, as I said, are more secretory and less phagocytic. So again, as phagocytes, they're pretty useless. But of course, they're secreting lots of colony-stimulating factors, prostaglandins, cytokines, etc., and upregulating inflammation. Now, the key thing really is that, and this will all start to sound rather familiar in terms of atherosclerosis, although the mechanism is different. In mycobacteria, when they're within the macrophages, they are thought, and we will come back to this as a pathogen mechanism as well, they are thought to change the metabolism of lipids within the macrophage. So they cause dysfunction in terms of lipid metabolism. And what this ends up causing is that a load of lipid starts to collect in the macrophage alongside the mycobacteria that's still alive, of course. So the macrophage starts to fill with lipid. Of course, in atherosclerosis, they were phagocytosing it. In this case, the mycobacteria that they phagocytosed are causing the macrophage to produce the lipid. And they become foamy macrophages. Again, lipid-filled, foamy macrophages. And of course, as we know from similar process in atherosclerosis, these are somewhat ineffective. So they're not good as phagocytosis. Instead, they're big, lardy, lipid-filled couch potatoes. And they're also very prone to apoptosis. So all these three mechanisms here, all these three possible changes to the macrophages, all actually make the macrophages less effective as phagocytes. So within our granuloma now, we have foamy macrophages. Foamy macrophages are full of lipid and are dysfunctional. We also have these epithelioid, slipper-shaped macrophages that are more secretory and less phagocytic. And maybe a few giant cells, fused macrophages that die quicker. And around the edge, we have our fibroblasts, all secreting collagen. So we've got our fibrosis, our walling off of these key cells within the granulome. So all well and good. Okay, we've got some live mycobacteria trapped inside macrophages and some dysfunctional macrophages, but it's all walled off in this nice little ball surrounded by fibrosis. But it can go a little wrong. So we end up with this caseous necrosis, and again, there's a lot of similarities here, isn't there, with atherosclerosis, because we're going to get this necrotic center, this necrotic core. The necrosis, again, is going to be due to ineffective efferocytosis. So our macrophages that do manage to undergo apoptosis result in our normal little nice packaged up sealed apoptotic bodies with the live mycobacteria in the lipids and all the rest of it. But because of course the surrounding macrophages are all, they're foamy cells full of lipid, they're more secretory than phagocytic if they're epithelioid cells, or they're giant cells and they're on the point of dying themselves anyway. They're not cleared by efferocytosis. So we end up again with these apoptotic bodies then going into secondary necrosis. So they eventually degrade. When they degrade, they're going to release live mycobacteria and lipid into the core of the granuloma. And of course, the lipid component of this gives us this cheesiness known as caseation. So we have caseous necrosis. This caseous necrosis, again, a bit like our atherosclerosis, it liquefies. And this can cause the granuloma, in this case, to cavitate. So in other words, the liquid will all drop to the base and we'll end up with this big hollow ball. And you can imagine this is going to make that granuloma a bit more prone to rupture. It's going to be more unstable. If it ruptures, of course, we're releasing live mycobacteria potentially into the circulation, and as a result, our latent TB becomes active TB. So our live mycobacteria and lipid, when we go through this secondary necrosis, are released into the granuloma. So we've got this mess of cheesiness, caseation, and live mycobacteria and dead macrophage. The caseous necrosis liquefies. When it liquefies, of course, it drops to the bottom of the granuloma and cavitates the granuloma. The granuloma becomes hollow. The granuloma then ruptures, releasing live mycobacteria, certainly into the surrounding environment, in other words, the lungs, but also potentially into the circulation. So the live mycobacteria can now cause more granulomas in the lungs or travel around the body and cause granulomas elsewhere. And this is where we were saying about, yes, you might be aware of TB of the spine or TB of the kidney, but originally it will have been inhaled. There will have been a granuloma in the lungs, albeit a small one, that then ruptured and released this live mycobacteria so it can travel around the rest of the body. So rupture releases live mycobacteria, which may cause granulomas in other parts of the lung, in various organs or bone, or more often than in children, and possibly because their connective tissue is not quite so well developed, it causes miliary TB. Miliary TB is where you have lots of small granulomas throughout the lungs, a bit like confetti. So there on the left you can see a cavitated granuloma. So you can see that nice round shape. In the middle we have a calcified renal granuloma. Remember we said before about atherosclerotic plaques can become calcified. And the mechanisms aren't totally understood about how this happens, but it can happen. And there on the right you have miliary TB. As I said, it perhaps looks a bit like one of those snow scenes that you shake up and the snow whirls around everywhere. So, as we said, latent TB may not show on chest X-ray. But, and this is quite logical really, people who do have latent TB, who do show some sort of lesion on the chest X-ray, appear to have a greater risk of developing active TB. And if you think about it, that's possibly quite logical because maybe it's a bit larger. So if it's larger, it's going to be more prone to rupture. Which brings us to our applied section. So we've talked about acute infections, whether it be from viruses, whether it be from bacteria. We've talked about TB. In your handouts before the session, you looked at asthma, COPD, and you've also looked at the pathogens that are going to cause our infections. So in this part, we're going to look at the signs and symptoms of respiratory disorders in general and what those mechanisms are. So you've got to start at the top, haven't you? Usually one of the first symptoms of certainly any sort of viral infection, other than COVID, we've got sneezing and a runny nose. Adenovirus, maybe rhinovirus, something like that. Okay, now this is the first time we've talked about the trigeminal nerve, cranial nerve 5. It's not going to be the last time. When we talk about headache, it's a key nerve involved in that, and one of these pathways will then look familiar again. So we have the trigeminal nerve. The trigeminal nerve is mainly sensory. It has C and A delta fibers. These A delta fibers innervate the nasal epithelium and the anterior nasopharynx. So there are sensory nerve endings. The receptors for these, we have chemosensitive ones. So these are chemosensitive to histamine via H1 receptors and various other chemical mediators. They're also mechanosensitive to particles or touch and potentially thermosensitive. If you think about it in the cold weather, well, I don't know about you, but in the cold weather, my nose always runs. So if enough of them are stimulated, so you can see what we're talking about here, we're talking about multiple EPSPs. They get summated at the trigger zone. This may generate an action potential. This action potential in the trigeminal nerve travels along, it goes through a ganglion, it enters the pons, and then it drops down into the medulla. And it goes to the sneeze center in the medulla. The sneeze center then stimulates motor efference to the respiratory muscles. Think about sneezing. You need to breathe in to the respiratory muscles, which causes inspiration and expiration. You don't get the compression of the glottis in a sneeze that you get in a cough, though. So you end up getting this mechanical sneeze. It also, the trigeminal nerve, through a reflex pathway, it stimulates parasympathetic efferents from the facial nerve. So these were afferents coming in, our motor efferents are efferents going out. Here we've got parasympathetic efferents. These are traveling out within the facial nerve. We'll look in a minute at a diagram explaining that connection. Because they're parasympathetic efferents, of course, they're going to cause vasodilation. When they cause vasodilation, in terms of the nose, and the glands as well, they cause hypersecretion, rhinorrhea, glandular hyperplasia, mucosal swelling, and lacrimation. So we get this facial nerve fanning out into the face, vasodilating due to a parasympathetic action. That vasodilation increases secretions, increases swelling. So I promised you a diagram. Here we go. So we have transduction, sneeze transduction in this case. We have chemical, mechanical, or thermal transduction of the trigeminal primary afferent that innervates the nose and the anterior nasopharynx. So an action potential travels in, travels to the medulla oblongata, to the sneeze center. It then synapses with a second order afferent that goes down to the respiratory center. That respiratory center, of course, stimulates our ventral respiratory group. We get inspiration, we get expiration, and we get a sneeze. But that second order also branches up to the pons above the medulla, to the superior salivatory nucleus, the SSN. We'll come back to this when we talk about headache. This superior salivatory nucleus is where that branch from the second order afferent synapses with parasympathetic efferents of the facial nerve. There's a ganglion somewhere in there, but don't worry too much about that. As a result of that, we have vasodilation and transudation, which gives us the runny nose, the runny eyes, the swollen mucosa, and all the rest of it, congested sinuses. So the parasympathetic reflex causes the initial clear coloured nasal secretions, because initially we're talking transudate. As we start to get an inflammatory process, we might have thicker exudate. If we've got a lot of inflammatory proteins, a lot of cells getting into the transudate, protein-rich exudate. And then we may get a yellow, fading into green the stronger it gets, colouring of this exudate. And this, of course, is caused by the degranulated or dying neutrophils, which contain the green enzyme myeloperoxidase, MPO. And that's what then colours it, these disgusting colours. Which brings us on to our next most obvious symptom, cough, but also mucus, because you can't really separate the two. Obviously, you may get dry coughs, but generally, if we're talking about an infective cause, you end up getting cough and mucus, or any long-term condition. So, of course, we can cough, we can make ourselves cough. So it can be voluntarily initiated by cortical control. But in terms of clinical presentations, it's caused by mechanical or chemical irritants within the conducting zone, within the larger airways. Below that, below the sort of smaller bronchioles, there are no active cough receptors. And of course, this has some significance in terms of if you start getting consolidation in the lung parenchyma, in the alveoli, it's not necessarily going to stimulate cough. Right, we have 3 different cough receptors. Well, we actually have 4, but one of them is not really a cough receptor. Our main ones are C fiber receptors. And you recognize C fibers, of course, from pain, from the trigeminal nerve we just talked about in terms of sneeze. It's a sensory fiber. C fiber receptors are chemosensitive in this case. We also have rapidly adapting receptors, which are mechanosensitive. And then on top of that, our other mechanosensitive ones are, imaginatively named cough receptors, which is a bit confusing because they're all cough receptors, but these are actually called cough receptors. Probably the later one discovered. These are also mechanosensitive, but they're mechanosensitive not to big things like bronchoconstriction. They're mechanosensitive to light touch, to small, tiny touch. So punctate stimulation. All of the receptors transmit action potentials via the afferent vagus nerve to the cough center, also in the medulla. The cough center then initiates the cough reflex via the respiratory center. So let's talk about chemical stimulation. So this, remember, is via C fiber receptors. They are the predominant sensory neuron in the lung, in the upper airways. If you remember, we talked about J receptors, which are also C fibers, but they're down around the alveoli, nothing to do with cough. They're all about the perception of dyspnea, nothing to do with cough. These C fibers in the larger airways are sensitive to chemicals. The chemicals they're sensitive to, things like bradykinin, protons, prostaglandin E2, nicotine as in the chemical, capsaicin and other chemicals. Capsaicin we've got in there because capsaicin is the hot component of chilies. And if you have ever been chopping chilies, you inhale lots of chili fumes, they likely to make you cough. They also make your nose run, make your eyes run, particularly if you put your finger in your eye, and they burn your skin. And they're not significant pathologically, other than being uncomfortable, but they are significant in terms of research on C fibers. So you will see quite a few studies that are doing studies on C fibers, and they will use capsaicin to identify C fibers because they all have receptors for them. So not pathologically significant, really, not unless you happen to chop a lot of chilies, but the others are. And if you notice, the others are all released in inflammation. Like all C fibers, these C fibers synthesize and secrete neuropeptides when stimulated. So obviously we have the potential here for that neurogenic inflammation. So our neuropeptides, a calcitonin gene-related peptide, This causes vasodilation directly by binding to smooth muscle cells. Substance P degranulates mast cells, which releases histamine. And of course, histamine is a vasoactive amine, so it vasodilates vascular smooth muscle, but it actually causes bronchoconstriction because it contracts bronchial smooth muscle. So different receptors, different effects, completely opposite effects. In one case, we contract smooth muscle in the bronchi. In other cases, we dilate it, as in the vessels. Histamine also increases vascular permeability and, as we said, contracts bronchial smooth muscle, causing bronchoconstriction. It also, when we talk in a little while about mucus, it upregulates the solid component of mucus, which is mucin. Neurokinin A, our other neuropeptide, also causes bronchoconstriction and recruits eosinophils. And as you saw from your asthma handout, eosinophils can cause a lot of damage. One thing that is interesting, it's really controversial that in anaesthetized humans, and I believe in dogs as well, our C fiber receptors, when they're stimulated, inhibit cough. And no one has really worked out why this happens. But generally, when you're awake, your C fiber receptors, if you stimulate them with these chemicals, it will cause you to cough. So mechanical stimulation. So our biggest group really of mechanoreceptors in this case are the rapidly adapting receptors, RAS. RAS are mechanoreceptors, hence they're mechanosensitive. Transmit action potentials via A delta fibers. That's really where the rapidly adapting bit comes in. They are sensitive to bronchoconstriction. And you can see what's happening here. Hang on. We just had C fiber receptors that caused bronchoconstriction. And now we've got rapidly adapting receptors that are actually going to make us cough because of that bronchoconstriction. We're starting to get a bit of a chicken and egg situation going. They're also sensitive to other mechanical stimuli like mucus, like edema, and they're also sensitive to punctate stimulation. So here, whereas our C fiber receptors were sensitive to nicotine, the chemical in cigarettes, in this case, the RAS are sensitive to particulates in the tobacco smoke. So a mechanical stimulation, so you get both chemical and a mechanical stimulation to cough from smoking. Cough receptors, as I said, it's a bit of a confusing name. They're all cough receptors, but these are specifically called cough receptors. They're localized version of our rapidly adapting receptors, so they're very similar. They tend to have, though, many free nerve endings. So this makes them quite sensitive to tiny, tiny things. So they're highly sensitive mechanoreceptors. They're found again in the upper airways, in the larynx, the trachea, the bronchi. And they respond to punctate mechanical stimulation, not big things like bronchoconstriction. If they respond to mucus, it'll be to tiny, tiny bits of mucus. As I said, there is another one, but it's not really a cough receptor. So you may see in articles about slowly adapting cough receptors, and they're not really cough receptors. What they are thought to do is detect stretch. If you remember when we looked at respiratory and physiology, we looked at the Herring-Breuer reflex. This was where if you try and inspire too deeply, it will stop you doing that, to stop you damaging your lungs, and it will cut you off with a cough. So these are not really pathological. These are about detecting stretch. They're also, conversely, thought to stimulate bronchodilation as opposed to constriction. So we don't need to worry about these in terms of pathology. All of these, as we said, we have the vagus nerve, goes to the respiratory centers, and it stimulates the cough reflex. The cough reflex has 3 phases. The first is the inspiratory phase. This is the inflation of the lung, which stretches the diaphragm and the muscles. That stretch gives us the optimum recoil so that we get the most force behind our cough. Then we get a pause. We get this brief 2 milliseconds, 0.2 of a second, closure of the glottis. We don't get this with sneeze though. We only with cough. And this closure of the glottis, what it does, it builds up the pressure in the lungs even more so that we can get maximum force for the next phase, which is, of course, the expiratory phase. So the glottis opens. The initial flow is known as the cough spike. This is a supramaximal expiratory flow for about 30 to 50 milliseconds, so 0.3 to 0.5 of a second. We get this really forceful cough spike. After that, you get a slower expiratory flow lasting for about 200 to 500 milliseconds. This dynamic compression of the airways during the expiratory phase, combined with ciliary action, sweeps debris and mucus up to the mouth for either expectoration or we swallow it. Swallowing it is a much cleaner way of getting rid of it because gastric acid destroys it, end of. Sputum, rather than sticking it in a pot or a tissue to show the nurse when you go and see her. So the cough reflex then, we've already sort of talked about this chicken and egg situation. You have this synergistic interaction of the receptors, and this helps to explain really why coughs can be so persistent. So, of course, damage to any respiratory epithelium due to any pathogens, anything like that, is going to cause inflammatory changes. So we get arachidonic acid cascade, so we get our prostaglandins, our leukotrienes, all the rest of it, platelet activating factor, mast cell degranulation, vasoactive changes, etc., etc. We also get potentially phagocytosis of damaged tissue or pathogens, which again leads to secretion of inflammatory mediators. These inflammatory mediators, bradykinin, PGE2, etc., all stimulate our CFRs, our C fiber receptors that are chemically sensitive. The other thing that happens is, and we'll look more about this in a minute, inflammation upregulates mucus, and it does it in 2 ways. One of the ways is it upregulates the solid component of mucus, mucin, and inflammatory exudates add to the volume of mucus. So we get mucus upregulation, we also get bronchospasm, And that's going to then stimulate our mechanoreceptors, our RAS and potentially our CRs, our cough receptors. There is also evidence that the activation of the CFRs can sensitize those cough receptors, the ones that respond to punctate stimulation, peripherally, so in other words at the receptor level, but also centrally at the synapses. And of course this is logical because we've got neuropeptides. Neuropeptides do tend to sensitize other receptors and do sensitize synapses. And of course, we've got the effects of neurogenic inflammation. So we get an upregulation of inflammation, which again activates the mechanoreceptors and the chemoreceptors even more. So it's no wonder really that coughs, once they start, can be quite persistent. As I said, we can't really separate coughs and mucus because they're again, we've got a bit of a chicken and egg situation going on here. So let's talk about mucus first. As I said, we've got two components really. And that gives us two layers. So we've got an upper gel layer that acts a bit like a raft being pushed along. Below that, we've got a watery sol layer that enables the cilia to waft through it and move that gel raft along. So the gel layer has a high concentration of glycoproteins called mucins. The sol layer is mainly water, and this allows the cilia to waft through it and move the gel layer along like a raft. Mucus as a whole contains various other things. So it contains ions, electrolytes. It contains enzymes, antibacterial secretions, obviously bacterial products potentially, chemical mediators, and cell debris. Mucus needs to be the right stickiness. It needs to be of an optimal viscoelasticity for the cilia to move it. That viscoelasticity, or the component that makes it sticky, are the mucins, and the mucins form about 2% normally of mucus weight. The more mucins we have, the stickier it gets. And again, MPO, myeloperoxidase, like with our nasal secretions, from neutrophils gives us that yellow to green colour, depending on the concentration, really, of the myeloperoxidase. So mucus volume can be increased by increasing the mucins, or by increasing the water content, or both. So here we're talking about the volume of mucus as a whole. If we increase the mucins, we increase the stickiness. So in this case, if you increase mucins alone, you've got very sticky mucus that's going to be harder for the cilia to move. And here you can see our cilia. They have this very coordinated way of moving through the sol and moving the gel, the mucin component, along up towards the mouth. And as we said, increases in the volume of mucus itself, so those two layers, can be caused by hypersecretion of the mucins, but also by inflammatory transudate or exudate. As we said, if viscoelasticity increases due to a high secretion of mucins, the cilia are going to have to work harder to remove it. The other issue here is that smoking and endothelial damage for any reason will shorten the cilia, which obviously is going to make them less effective. You get cilia blunting. Smoking and endothelial damage also reduce the frequency and coordination of beating. So they are not working together as effectively to sweep that mucus out. So are mucins then? As we said, mucins are glycoproteins. They also have on them broad-spectrum pattern recognition receptors, a bit like our phagocytes. If you remember, PRRs bind to PAMPs and DAMPs. The PRRs in this case aid pathogen attachment or debris attachment so that this is removed with the mucus. It adds to the attractiveness to anything that we want to get out. Mucins themselves are coded for by 9 MUC genes. And this is where really you have quite a broad potential for mutation in those genes and alterations in mucus secretion or mucin secretion, which is going to change the viscoelasticity of your sputum. Mucins are synthesized then by goblet cells. I'm sure you've all heard of goblet cells. Goblet cells sit between the epithelial cells of the respiratory tract. And you can see one there in the diagram. It opens directly into the lumen. But also, mucins are secreted by submucosal glands in the respiratory tract. These are much larger glands that sit below, and within them they have cells that synthesize and secrete mucins. Those mucins are then released into the gland, move out of the gland into the lumen. When they are released, mucins are a bit like those flannels and towels and things you get that are packed up really, really tight until you open them or wet them. And then all of a sudden, they go poof, and they really, really expand. So as soon as they're exocytosed, mucins expand humongously to fill up the space. So how do we increase our mucin secretion? Well, in the short term, if you look at this list, there's an awful lot of inflammatory mediators there. So we've got proteases. Proteases increase mucin secretion. We've also got cytokines. And note the first three: TNF-alpha, interleukin-1, interleukin-6. The big bad boys that come out to play in chronic inflammations. So COPD, tuberculosis. Interleukin-13 is common in asthma. Some leukotrienes increase mucin. Substance P, so one of our neurokinins, also, apart from degranulating mast cells causing bronchoconstriction, also increases mucin secretion. Nicotine, the chemical nicotine, which, if you remember, stimulates C fiber receptors to cause cough. It also binds to our goblet cells, submucosal glands, and increases mucin secretion. ACH is another one, bradykinin and histamine. So not only do they cause bronchoconstriction, they also increase mucin secretion. Platelet activating factor is an inflammatory mediator we talked about way back in immunology. Comes from most of the leukocytes and also endothelial cells. And this is an interesting one, shear force. We're talking about a mechanical event that increases mucin secretion. So a mechanical event here could be cough or could be heavy breathing. So if you cough, the mere act of coughing will potentially make your mucus stickier. So we've got this bit of a cycle going on again. We've got inflammation stimulates cough chemically via those mediators, but also mechanically via mucin upregulation and inflammatory exudates. Both of these, of course, are going to increase mucus volume. The more mucus we have, then the more we're going to stimulate the mechanosensitive cough receptors. Cough itself also upregulates mucin secretion. So we've got a bit of a cycle going on. In the longer term, so this is about short-term upregulation of mucin secretion. Yes, we've got more mucus or stickier mucus. In this case, we've got a long-term chronic condition. So if you keep stimulating, what happens is that epithelial cells between the goblet cells go into metaplasia. They change. They become more goblet cells. So you have fewer epithelial cells, more goblet cells, more mucin. They also get larger. Our submucosal glands, also you will get more of those and you will get them larger, so more productive. So both the goblet cells and the submucosal glands become more chronically able to produce more mucin. Which brings us on to hemoptysis, really, because, well, yes, obviously you don't always have hemoptysis, but in some conditions, when you bring your mucus up with your cough that we've just been talking about, there will be some blood in it. So why is that happening? One of the things is angiogenesis. All tissue repair, and bear in mind if you've got a chronic lung condition, you've got ongoing tissue damage and repair. If you've got tissue repair, you need a new blood supply to supply those new cells. And this is where you will bud off little capillaries to grow into that new tissue to supply it with nutrients. So you get the growth of new, fragile little buds off existing capillaries. This is usually stimulated or primarily stimulated by vascular endothelial growth factor, but also to a certain extent by fibroblast growth factor. And of course, these are very fragile little new buds. Because they're fragile, they break easily and bleed. So if you've got shear force, you're coughing a lot, that's going to break them. So, and this is even more likely in chronic conditions because you have this tissue damage going on at the same time. So you have proteases, you have reactive oxygen species, lysosomal enzymes, all sorts of things, cytolysis, apoptosis, necrosis, all damaging tissue. So potentially damaging these fragile little buds from the capillaries that are trying to grow into the healing tissue. So particularly common in chronic conditions. In terms of proteases, the prime example we usually talk about is metallomatrix protease 1, MMP1. And this is definitely increased in a lot of chronic respiratory problems. It degrades the extracellular matrix. Now, If you're trying to proliferate cells into an area, that's really useful. But in this case, it's gone a bit crazy and it's starting to damage the delicate vasculature. In addition to this, you've got some mechanical effects in terms of TB. You imagine you're forming this granuloma. This granuloma can rupture fragile vessels, or it causes vessels to have to divert around it. So they become weaker and engorged and tortuous and more prone to rupture. Of course, it's an inflammatory condition, so you'll have vasodilation as well. In tumours, and we'll look at this when we look at colorectal carcinoma, tumours are very good at stimulating angiogenesis, even more so because they have all these mechanisms to enable them to survive, to proliferate. So they're very good at stimulating angiogenesis. So any cancer will likely result in some sort of bleed. And because we're talking about respiratory conditions here, of course you're going to see that bleed in terms of what's being coughed up. Okay, you can probably work all of this out for yourself. We're going to have shortness of breath. We're going to have potentially increased respiratory rate because, of course, we're going to have a certain element of obstruction to airflow. And if we're talking about consolidation or fluid in the alveoli, to gaseous exchange itself. So mucus, exudate, and pus can all inhibit airflow and gaseous exchange. And in more chronic conditions or more significant conditions, things like emphysema, collapsed airways, bronchoconstriction, fibrosis over a long term can reduce airflow and gaseous exchange even further. We're decreasing elasticity, etc., recoil, we can't refill the lungs. So lots of issues there with airflow. Of course, what happens is our central and peripheral chemoreceptors, which will respond to a high CO2 via monitoring H+, or very low hypoxia, and in response to that, they will increase respiratory rate and depth via the respiratory centres. And then we've got to think about our J receptors. As we said, they're located in the alveolar walls next to, juxta, the capillaries. And they're thought to cause the sensation of dyspnea in response to vascular engorgement or interstitial edema, which could be caused by a variety of things. Could be a PE, could be pneumonia, pulmonary edema. In addition, there may be an element of anxiety and also this awareness of difficulty breathing due to being aware of the effort of the respiratory muscles in expanding or contracting, relaxing the chest. So we get a feeling of dyspnea driven by the sensation of tightness. That tightness could come from awareness of the respiratory effort, from anxiety, or due to the stimulation of the CFRs in the larger airways. Chest pain. So we've got two real sort of types of chest pain, haven't we? And we often in clinical practice will ask about what sort of pain they have and where it is. Pleuritic pain is mainly because there are nociceptors in the pleura. There's none in the parenchyma of the lungs, but they are in the pleura. So these nociceptors in the pleura are most likely to be myelinated because it tends to be a sharp pain. So therefore we're assuming we've got a fast transmission of the action potential, conduction, is rapid. So they transmit action potentials rapidly, so we get a sharp pain, tends to be very well localized, and of course it tends to occur on inspiration and expiration, because what's happening is we've got the two membranes rubbing together. So we've got friction and we've got potentially inflammation around the pleura. And then we have the other type. We have the one where you'll get someone, you have some pain? Yes, I have chest pain with my cough. And they put their hand, don't they, over the center at the top of their chest. Oh yes, it's burning here. So that retrosternal burning pain is most likely due to the bronchial nociceptors, probably CFRs and RAs, that are sensitized by the release of the neurokinins. And that gives that burning retrosternal pain. So, not a sharp pain because, of course, we're talking unmyelinated. The other thing is if someone feels short of breath, or particularly it's something we've got to think about in these days with COVID, this idea of anxiety. You imagine someone comes in chest pain and a cough at the moment, they're going to be quite anxious. So anxiety enhances pain perception. So we've got to bear in mind that add another layer on in terms of the patient's perception of the pain and what it means to them. Okay, get your stethoscope out. You all know these sounds anyway, don't you? I would hope so by this point. Okay, so a wheeze. A wheeze is going to be down to bronchoconstriction. This bronchoconstriction, if you remember, could be due to histamine, leukotrienes, PGD2, PAF, substance P, various other things. We may have additional obstruction because there may be edema around the cells, so that's going to reduce the lumen. We may have mucus blocking the airways or partially blocking the airways, giving us a wheeze. Inspiratory wheeze is supposed to indicate an upper respiratory tract obstruction, expiratory, smaller airway, so lower down. Biphasic, you've got problems. Stridor is a form of wheeze, but here we're talking about a much higher up obstruction. So we're talking about laryngeal or glottal obstruction. Crackles and creps are really quite cool. We've got multiple small airways and alveoli. So they collapse and then they pop open as the airflow changes, or they manage to overcome their stickiness, and they suddenly pop open and you get that little popping sound in the bases. Rhonchi, I like rhonchi. Rhonchi always sound like snoring. So you've got this low-pitched, continuous sort of wheeze, and that usually indicates some sort of obstruction by viscous mucus. So viscous mucus, of course, is going to have a higher mucin content than an exudate content. Okay, so that brings us to the end-ish. If you haven't done it already, or if you can't remember what it was about, please go back and have a look at the unwell person handout in the module preparation section. So that in there we're talking about sickness, behaviour, anorexia, fatigue, cachexia, fever, that sort of thing. The sort of presentation that can come alongside any chronic condition, particularly a chronic inflammatory condition, whether it be respiratory or not. Hence, it's in the module preparation section as opposed to in the respiratory section. But if you can't remember what it's about, please have another look at it. In there, we're talking about our big bad boys, TNF alpha, interleukin 1, etc., etc., and the impact they have on the systemic patient with any particular localized condition, chronic condition. So they are key signs and symptoms, really, that cut across all the systems. So please go back to that if you can't remember it. If you haven't looked at it, now's the time to look at it. And of course, the usual. There is a handout for this one, for this video. Make your own notes and diagrams. After every single topic, learn the topic using the techniques that you used in physiology and hopefully using a study buddy. If you find it's not making sense, you can't remember what a respiratory membrane is, I would hope you could, then go back to your physiology and refresh your memory. And please don't put this off, because we still have 3 more topics and you have a mock exam looming up very soon.